to secrete proteins. They secreted proteins which were synthesized on ribosomes found in the endoplasmic reticulum, which of course suggests that the ER functions to target proteins to the surface of the cell for secretion. And with a little bit of goading, you also recognize that these things synthesize proteins that are integral to the membrane, so integral membrane proteins that transverse the membrane, or at least are partially included in the membrane. And so what we're going to do today, and see how far we get, is to discuss targeting of proteins in cells. How do they get where they're going? So a protein is made out of ribosome and the cytosol, and up to the cytosol. What about all the other proteins in the cell? What about proteins that go into mitochondria and chloroplasts, or proteins, as I said, that are in the membrane? Proteins that go into lysosomes, proteins that are secreted from cells. How do they get there? How are they trafficked? So it's a question of trafficking. And this sort of illustrates that to you, that there are all these different compartments in the cell: mitochondria, the nucleus itself, like you mentioned, the nucleus, chloroplasts, lysosomes, the peroxisomes, similar peroxisomes, and all of these things contain proteins. How do those proteins get there? A lot of these are organelles. They have membranes, like the lysosome, right? Some of them have a double membrane. How do proteins get into the ER? So the ribosomes attach, but how do the proteins get into the endoplasmic reticulum? How are they targeted to their wherever they're going, to their destination? And this is a little figure that you should refer back to as you're studying, because it shows several things, and that is that primarily, you notice that we start with cytosol, which is where protein synthesis occurs. All protein synthesis begins in the cytosol at least. And then proteins from the cytosol are targeted into the nucleus, into organelles like the ER, the mitochondria, and the peroxisomes, into other organelles such as the Golgi, into lysosomes. Some are secreted, some end up going just to the surface of membrane proteins and so forth. How do they do that? And this chart describes that there are three ways. Proteins move into the nuclei of cells through what we call gated channels, gated transport channels. There are actually nuclear pores or channels or gates through which proteins can move into the nucleus. And that's the easiest one to understand or think about. Proteins also move through membranes. They move through in a process called translocation or transmembrane transport. And the proteins that we talked about that are synthesized on ribosomes attached to the endoplasmic reticulum. So ribosomes there attached to the ER, and proteins move into the ER by this process here, transmembrane translocation or translocation. And then finally, there are proteins that move throughout the cell, either from, not either, but including from the endoplasmic reticulum to the Golgi, and from endoplasmic reticulum to lysosomes, from endoplasmic reticulum to secretory vesicles to the surface of the cell, and they move in vesicles. They move through the cell in little tiny vesicles. We'll talk a little bit about that. What we're going to do today is we're going to concentrate primarily on this step here. We'll talk a little bit about nucleus, a little bit of mitochondria, but mostly we're going to talk about this process by which secretory proteins get from the cytosol into the endoplasmic reticulum, how they translocate the membrane. This is just a cartoon to show you, we'll talk about this later, about vesicular transport, which is the one that was in this picture that was shown in green. So all of this is vesicular transport. And then the vesicular transport process is actually, the process actually includes the budding. So where's the target? Here's the donor compartment here, right? It includes the budding of materials inside that from the donor compartment into a membrane vesicle. And that membrane vesicle is moving and fusing with another set of vesicles to target. And this is the process by which proteins move from the endoplasmic reticulum into the Golgi apparatus. So we'll talk about that, but not today. I just wanted to let you know what that was. So how do proteins get into the membrane, into the endoplasmic reticulum? Well, the vast majority of proteins that get into the ER get in there, and certainly all of those that are soluble in the ER, the soluble proteins, non-membrane proteins, get into the ER because on the end of the protein there's a region which is known as the signal sequence or the signal peptide. And the signal peptide is a stretch of amino acids that contain somewhere between 16 to 26 residues. So this is the signal peptide, all these colored residues. And that signal peptide somehow identifies the rest of the protein, the entire protein that's synthesized, as a secretory protein or some sort of a protein, a lysosomal protein or whatever, a protein that needs to get into the endoplasmic reticulum. Those amino acids are not homologous, at least not in terms of amino acid sequence. They are analogous in terms of their structure. For instance, as it says here at the beginning of all of these, there's a basic region, basic amino acids. And most importantly, there's a stretch in the middle of anywhere between 7 and almost a dozen, and very slightly, amino acids that are hydrophobic residues. And those hydrophobic residues are essential for getting this into the endoplasmic reticulum, and I'll tell you why. And then at the end there are some residues that are non-helical that are followed by a small either glycine or a proline. And this region here, which is a non-helical region followed by either gly or pro, is a sequence which indicates to an enzyme that this sequence can be cut. So you can see there's a cleavage site there. The cleavage site is preceded by glycine and the proline. This just tells you what I said, that movement through is the term translocation. You should know that word. And that secretory proteins are first translocated into the ER, which leads them to other compartments in the cell. And this is an important statement because it's one of the major functions of the endoplasmic reticulum. And that is that the endoplasmic reticulum is a targeting organelle. It targets proteins for different locations within the cell. And we'll look a little bit about that. So how do proteins get in there, into the endoplasmic reticulum? How is the protein that's being synthesized targeted to be included in the endoplasmic reticulum? All protein synthesis begins with the cytosol. So even though we saw free ribosomes, and we said those are proteins being synthesized that will stay in the cytosol, proteins that don't stay in the cytosol still begin synthesis there. So the difference is whether or not that ribosome, once it begins synthesizing a protein, whether or not it stays soluble in the cytosol or attaches to the membrane. And the reason that it attaches to the membrane, or the way that it attaches, is due to the signal peptide. Remember, we talked about that 26 amino acid. And that 26 amino acid sequence is recognized by what we call the signal recognition particle. So the signal recognition particle recognizes that guy and binds to it. That signal recognition particle, this is just another cartoon that shows the same thing. Here's that SRP, signal recognition particle. That signal recognition particle is a ribonuclear protein particle. It has ribosome, has proteins and RNA in it. But it recognizes this sequence. Remember when we talked about ribosomes, we got the large subunit that actually had a channel or a pore that came out of it. That pore contained about 25 or so amino acids, or it took 25 or so before it started to come through. So here's this thing that's 26 or so. As this comes through, the signal sequence comes on and the SRP attaches to it. This SRP has two functions. One is to direct this ribosome that has the signal peptide to the membrane, to attach it to a membrane. And the second function is that it stops protein synthesis. So when the SRP attaches to the nascent peptide, right? This is a nascent peptide being synthesized. When it attaches to that signal sequence, protein synthesis stops. Why does it stop? You can tell me why it stops. Well, go ahead.
I was just going to ask, but I might be wrong. The protein can't come out anymore?
So it can't come out anymore? That's right. So well, why don't you want it to come out anymore of that ribosome? I got to tie my shoes. Why don't you want it to come out?
Wouldn't it be easier if it was a single strand rather than a folded piece of protein?
The protein, when it goes through that channel, has to go through unfolded, right? It must go through as it's being translated. What we call that is co-translational translocation. So it's translocated while it's being translated. Translocated while it's being translated. If it's not, if it didn't stop, and continued to come out, the protein continued to be synthesized. Remember we talked about folding, we said that as the protein comes out, as soon as it does, it begins to fold. And if this protein folds, then it loses access to the translocon through which the protein moves. So yes, it stops its own fold, binds it to the protein, and then directs it to this translocon, this translocator. The way it does in this picture, in this cartoon here, right, is it binds to this structure, the orange structure there, they're calling the translocon. And the translocon are actually proteins in the membrane through which these proteins travel. At one time, we thought it was just something that the ribosome bound to. In fact, it was first called riboforin. And then they started to isolate these theory that the protein now is actually called the SEC61 translocon. And it's called SEC61, SEC, SEC61, because of the way it was found. These guys were identified in yeast mutants. So when people started to study this whole process, what they did is they took yeast cells and they exposed them to some sort of mutating reagent, either chemical like that photourea, or to something like UV light. They shot UV light. But they mutated these cells. They aimed to have a single mutation in the DNA per event, per cell. And then they looked at these cells, and first of all, what they did is they isolated cells that were no longer able to secrete. So they looked for cells that they had induced mutations in, they could no longer secrete a protein. And they knew that they messed up the secretory protein pathway. And so then as they found these mutants and they realized that they were all different proteins, they started to number them. SEC61, 62, or SEC1, 2, 3, 4, and so forth. So any of the proteins that we talk about in this pathway are proteins that inhibit secretion, and even a lot further steps. But anyway, this was the 61st call found, so it's not a molecular weight. It's a number that they were isolated in these secretory mutants. People understand that? These SEC mutants. So this is the SEC61 protein. This cartoon, which is from your book, leaves out another thing, and that is that this, I guess they show this interacting with the SEC protein, but actually the SEC protein, or the SRP, right, this is the SRP, actually interacts with a receptor. So part of that SEC complex is a receptor protein. This is the SRP receptor. The signal receptor protein receptor recepts that protein, and it binds to it, and it locates that to the membrane. Right? So when this binds to this, it locates to the membrane. And what this does is it interacts close to this translocon, the complex there, and it begins this thing to, once it does, then these guys come apart. Right? And this continues to be synthesized, and as it's synthesized, it's translocated. It's co-translocated, which means it's as it's synthesized, it's moved into the endoplasmic reticulum, and the protein moves in. What else can I tell you? Notice here that in this translocon, even in this cartoon, and this is pretty old, this is from an older cell biology book, but if you look at this translocon here, it has a plug in there. Right? It says it has a plug. I think they call it a plug here. And in fact, we now know that there is a plug, and there's a very good reason for that plug. Can anybody imagine why, when these translocons sit in the membrane, right? So this is the endoplasmic reticulum membrane. And here's this translocon sitting in there, and it's plugged. Why is it plugged?